All right, so it's a pleasure to introduce our speaker today. He's a professor of cognitive and neural systems at Boston University and a faculty member in the Speech and Hearing Bioscience and Technology program of the Harvard-MIT Division of Health Sciences and Technology. He's also a research affiliate in the speech group at the Research Laboratory for Electronics at the RLE at MIT and a visiting scientist at Massachusetts General Hospital. He got his bachelor's and master's degree in electrical engineering from Missouri and Princeton University, respectively, and then a PhD from Boston University in cognitive and neural systems. He describes himself as a computational and cognitive neuroscientist with interests in speech production, articulatory physiology, and sensory motor control. Please welcome Professor Frank Guenther.
First, thanks for inviting me, and it was great to talk to people and learn about what's going on here earlier today. I'll be talking about work that was done in collaboration with a number of people. I want to get this out of the way first because everything I'll be talking about was in collaboration with some of these people. This group of people are graduate students and postdocs in my lab at Boston University in cognitive neural systems. And these are outside collaborators. Most notably, we've done a lot of collaboration with Joe Perkel at MIT's Speech Communication Group, Shinji Maeda, Philip Kennedy, and Xavier Lario, and I'll talk about projects related to all of those guys as we go. The research that we do at our lab has as its primary goal the elucidation of the neural processes underlying speech, and we focus on speech production, and we look at learning of speech in children. We look mostly at normal speech in adults. And finally, we look a lot at breakdowns of speech in communication disorders such as stuttering and apraxia of speech. And we use a number of different techniques. First of all, we use a lot of neural network modeling. So we mathematically model the brain structures that we're looking at. We use functional brain imaging, in particular fMRI, and I'll show a couple of fMRI experiments today that helped us guide development of the model in our lab. And finally, we use motor and auditory psychophysics, including studies of articulator movements using articulometry and auditory trajectories, for example, of the produced speech in the model. And the studies that we do are organized around a modeling framework which we refer to as the DIVA model. And this is a neural network model of speech acquisition and production which we developed in our lab. In the bulk of today's talk, I'll be describing the DIVA model and how we test it and how we use it to look at communication disorders. So I'll first give an overview of the model and talk about learning in the model as well as extracting fMRI activity from model simulations. Then I'll talk about two major components in the model. These are the feedback control subsystem and the feed-forward control subsystem. And then I'll end the talk with a description of a use of the model to investigate communication disorders. In this case, I'll look at a case where we're investigating neural prosthesis for restoring speech to locked-in patients, guided partly by our model. So this slide shows an overview of the model. This is a full schematic of the model. And each of these boxes corresponds to a set of neurons somewhere in the brain. These larger boxes are cortical representations, and the smaller boxes are subcortical representations. And each of the boxes has been associated with a particular region of the brain. And this has been done over many years of looking at existing data and also some of our own studies that I'll describe. But for example, we have a representation in the ventral motor cortex of articulator velocity and position. In the auditory cortical areas, including Heschel's gyrus and the posterior superior temporal gyrus, we have an auditory target map, an auditory state map, and an auditory error map. In the somatosensory cortical areas of the ventral somatosensory cortex and supramarginal gyrus, we also have somatosensory target state and error maps, and so forth. We have a supplementary motor area in the model, and we have the highest level in the model, which is the left ventral premotor cortex. This is meant to correspond to Broca's area and the premotor cortex that's immediately posterior to that on the left side. We'll refer to that as the speech sound map in the model, and I'll talk a lot about its properties as we go along. This complete model is actually a little too complicated to work with for purposes of the talk, so I'll be using a simplified version of the model, which I'll talk about here. And the first thing to note is that the output of the model is a set of articulator positions that go to an articulatory synthesizer. Usually we use the Maeda articulatory synthesizer, and this is basically a set of, it's a piece of software that takes as input the positions of the jaw, tongue, and lips. It creates an area function of the vocal tract from that, and it can use this area function to synthesize an acoustic signal that would result from that vocal tract shape. So what the model does is it learns to move these articulators in order to produce a sound that matches sounds that we try to teach it. And it does this by first tuning up many of the mappings in here that are shown by arrows. These are mappings between different neural representations, and I'll talk about that learning process in the next few slides. So the output of the model is the articulator positions. Those generate sound from the Maeda synthesizer. We feed the sound back to the model. We also feed the model some somatosensory information about the state of the vocal tract. And these combinations of information are used in the babbling cycle, so the model can learn different mappings between the different representations, and then later use those learned mappings to help it produce sounds that you teach it. This video will give you an idea of what the Maeda articulatory degrees of freedom are. What I'm going to do here is just randomly babble the 7 articulatory degrees of freedom. And this is the first learning stage in the model, where we randomly activate the articulators that create sounds, and we learn the mappings between sounds and the corresponding articulator movements. And so here's what it looks and sounds like when we do random babbling with this model. Do that one more time. So you get a rough feel for the degrees of freedom in the model, and you can tell that it's not speech-like at all, the sounds that are coming out when you do this random babbling. The noises, they sound a little bit like vowels, but you don't hear any consonants that are properly timed and anything like that. And the goal of the model is to learn how to produce those sounds, and in particular produce a set of sounds that we present to it. So the first learning stage in the model, as I mentioned, is a babbling stage where we randomly activate cells in our articulator maps. That drives movements of the speech articulators. We get the auditory feedback and the somatosensory feedback coming back to the model. And the combination of these 3 forms of information is used to tune up these mappings shown in red here, which are mappings from somatosensory and auditory errors into corrective motor commands. And these mappings are very important for the feedback control system of the model, which is this blue block over here. And the feedback control system consists of 2 subsystems, an auditory feedback subsystem and a somatosensory feedback subsystem. And both of them are tuned, these mappings between these sensory spaces and the motor space during this initial babbling process. So we pick a few thousand random articulator configurations, move it around a little bit. That creates training data with sounds and the corresponding movements, and we tune up those mappings from that. Then the next stage of learning in the model is where we teach it specific sounds from a language. And this process is shown by tuning up the arrows shown here. The speech sound map in the model, this highest level in the model, is the input portion of the model. And what happens is that we'll activate cells in this map, and these cells correspond to different speech sounds. And in the model, a speech sound could be a single phoneme, it can be a syllable, and it can even be a short phrase. As long as it's given to the model as a chunk, it'll learn that as a single speech sound, and it'll represent that sound with a cell in this map. Well, what it has to learn is, when I activate this cell, what motor commands do I read out in order to properly produce the speech sound that I'm trying to read out? And that's what it'll learn in this imitation process. It's also learning an auditory target for that sound. We give the model a sound sample, which is treated as an auditory trajectory. So we'll have the first 3 formant frequencies across time. That represents a target. I'll show an example of that in just a second. That's stored in this set of synaptic weights. And eventually the model also learns a somatosensory target for the sound, which is, what should it feel like when I produce this sound properly? What kind of tactile information should I get back from my vocal tract, etc. So the tuning of these 3 mappings takes place in this imitation process, and the process takes place as follows. First, we present the model with an auditory target that we want it to learn. This is an auditory target for the syllable ba. And what it shows is a range of values for the first formant frequency across time, second formant frequency, and third formant frequency. And so what this is a time-varying region in the 3-dimensional formant frequency space. So each sound is some sort of time-varying region in this space. And that's stored in a set of synaptic weights that project from the speech sound map cells in the ventral premotor cortex over to the auditory error map, where we're going to compare this target to incoming sounds when we attempt to produce the sound. Okay, so stores that target. Then the second step in the imitation process is that the model attempts to produce the sound in order to tune up its feed-forward command. So it needs to practice producing the sound several times. It's a new sound, so it doesn't have some built-in pre-learned feed-forward command that it can read out. Instead, it has to attempt to produce the sound. This will generate errors. An auditory feedback system will hear the errors and send corrective motor commands. So we'll get an initial production which is largely under feedback control. Feedback control is slower and a little sloppier. It won't sound as well. But with each production attempt, the commands that were sent in by the feedback system on the current attempt will be added to the feed-forward command for the next attempt. And what that means is that the next attempt will be slightly better, and each attempt gets better because corrective information is being added in from the feedback system and being stored in the new feed-forward command. Yes?
How do you compare? What are you comparing the auditory space?
We are comparing incoming auditory information represented in terms of these formant frequencies.
So mean square error on the format estimation?
Basically. So the way it works, it's in a neural network, but the way it works is that if my first formant frequency, for example, is here, it's outside of this target region for this point in time. So what it'll do is it'll generate an error signal that says F1 needs to be decreased. That error signal in auditory space is then mapped into the motor commands.
Formant trajectories is really what you're comparing.
Right. So the model's learning to recreate formant trajectories by commanding the articulators.
So if you could just go back to the previous slide for a second. Yeah. So what's happening is that the mapping from the all the way down from the articulatory control signal that comes out to the auditory signal that is produced by the mouth. So this is the actual production. So this mapping somehow needs to be understood in the feedback control. Because when you see a difference in so let's say that, the line that goes from the ear into the auditory error map,
Okay?
that line basically is the observed or the heard formant frequencies.
Right.
And
those are
it's compared to the target formants, and you just generate an error signal that says this formant is lower, that's higher,
and then
that's
it
okay.
corrects it over here.
And then what signal to produce? That's the trick, right? Because you need to know how to control the...
Right. So what happens is, this is jumping ahead a little bit, but so I've got my auditory target, I've attempted to produce it, I hear what it comes back, I generate an error because my feed-forward command, and this arrow is the feed-forward command, right? It reads out the set of articulator positions. Well, the corrective command adds on to the motor command output, and that adjusts these weights so that they now subsume that. So by hearing the error, you're adjusting the motor command output, and you're storing that new command in the feed-forward command for next time. So you tune the feed-forward command using the auditory feedback system to slowly guide you.
So the mapping from the command to in the MIDA synthesizer, is it obvious? In other words, how to change the parameters?
How to change these articulator parameters to correct? No, and that's what the model is learning in this first babbling cycle is how do you go from the formant frequencies into the articulator positions? And so that's one of the main things it learns. And that was in the previous learning stage. Okay, so in the imitation stage then, we gave it a ba, it stored that in those weights right here. It tries to produce the ba with its existing feed-forward command. It's no good because this is the first attempt. And so there's an error signal that adds on activity here, which corrects the error, but it also updates the feed-forward command for the next attempt. And so this happens one step at a time, and each time the model gets a bit better with production, and I'll give you an example of this process here. So first you'll hear the sound target, in this case, that we gave to the model. In this case, it's an entire phrase. I don't know how well you can hear that, but...
Good job.
So that's the target. We extract the formant frequency trajectory, which looks like this for that target. And this is the model's first attempt, and this first attempt is entirely under feedback control because it has no feed-forward command. It's not too bad, actually, because the feedback control system was tuned up pretty well, so it actually is able to do it, but it's kind of sloppy. The movements aren't quite right. This is the second attempt. You'll see it's a little bit better. sounds a little bit cleaner.
Guenther.
And then this is the 6th attempt, and at this point, the model's pretty much fully learned how to produce this sound.
Guenther.
So here's the original,
Guenther. Guenther.
and that's the replication. If we look at the formant trajectories, we'll see that these are the model's formant trajectories after 9 attempts in this case. They look very much like the target formant trajectories, but the intermediate steps don't. And for example, here we see a case where there's actually an overshoot of the formants and then it comes back down. This is because of the delay in the auditory feedback loop. It takes about 100 milliseconds for information in motor cortex to make its way out and then back up to the auditory cortex. And so a corrective command can't happen until a good 100 milliseconds after the error occurs. And so here's a case where the error occurred and then the model corrected by pushing these down. So it was overshooting in the wrong direction and then correcting. And we can see in general that these trajectories lag the target trajectories because of this auditory feedback. But this gets better and better because more and more of the job is being done by the feed-forward controller with time.
So how have you learned the pitch trajectory of this?
Pardon?
How have you learned the pitch trajectory?
Well, the pitch, what we do in the model is we just mimic the exact pitch trajectory that we get in the target right now. So we're not doing pitch control, really.
What about the sounds that don't have formants like that?
Sounds that don't have formants like that? Well, it's kind of complicated, but for here, for example, in these stop consonants, we have what we call a virtual formant target. It's actually a formant target that would require you to push your tongue beyond the edge of the vocal tract to reach it. You can't actually reach it, of course, because no sound comes out, but this is a way you can specify closure of the vocal tract with a formant target. So even though there's no formants here, the model's moving toward a target and then back out during that time period. And that target's kind of like a virtual formant frequency. If that makes sense.
And because you copied the pitch, there is no sound over there because basically the pitch disappeared. So
During this part, yeah. Other questions? You all set? Okay. All right, so that's how the model learns a new word. And then once it's done this a bunch of times, it's practiced the word, it's stored forever. That's a syllable or a word that the model knows how to produce. And then we'll look at its kinematics, for example, of the productions. I mentioned that all the components of the model correspond to parts of the brain, and in fact we've taken every cell in the model and we've given it a stereotactic coordinate in the three-dimensional space that's often used for MRI analysis, and this is the MNI space. I don't know if any of you do any brain mapping work, but MNI space is just a three-dimensional kind of standardized brain space. So a three-dimensional coordinate corresponds to some position within the brain, and we've given all of our cells such a coordinate, so they're effectively embedded in a brain, and we can plot the results of simulations on this brain by showing what cells are active during the simulation. So this is quite similar to what you would get from an fMRI experiment, and that way we can compare the model's outputs to what we get in fMRI experiments. And so what we...
So the MIADA model has about eight controls.
Yep.
7 or 8?
7 degrees of articulatory.
Okay, 7, and so you have 7, okay, there.
Along here there's 7, and there are actually a couple for the voicing as well, but the 7 main articulator parameters are spread out. There's a motor representation and a somatosensory representation for each one along here.
And then you have 8 maps. What are the other ones?
These are other components of the model, so these are the auditory error cells that I mentioned, somatosensory. What's that?
The outputs from the box.
Or the cells that are inside of those boxes, actually, is what these are. Okay? All right. So the trick here is to generate a hemodynamic response. fMRI measures blood flow in the brain. The blood flow tracks activity in the brain. So if a set of neurons is firing for 100 milliseconds, say, what happens is the blood flow kind of slowly builds up in that area and goes back down. It actually takes several seconds for it to build up. It's much later than the activity of the cell itself. It's a slower version, and so to mimic that, what we do is we convolve our cell activities, which correspond to the electrical activity of a neuron, with a hemodynamic response function, and there are standard functions that come with fMRI analysis software, and that's what we use. So we construct a brain volume, and we show the results of a simulation. So here what you'll see are the brain activities that arise when the model says AB in this case. You'll hear the AB first. That's during the actual production, and then later you'll see the blood flow build up and then fall back off with time. So we can see basically that we get blood flow in these areas, and we could take a snapshot of that right at the peak of that blood flow, and that we can compare to the snapshot sort of thing that we get from an fMRI study. All right. Okay, and we've done a number of such studies, and in fact, with existing fMRI data, we use this data to help us localize the locations of the model's components when possible. And then, so here's a slide showing the model producing a set of CV syllables, and here's an fMRI study where humans did that. This is actually an older version of the model. You can see it matches pretty well to the activities we see here in the auditory areas, the motor areas. There are a couple of discrepancies, most notably here, the supplementary motor area is shown in these subjects. This was an older version of the model where we didn't have the supplementary motor area in it, so there was no activity there. But for the most part, we get a reasonably good match, and we've actually tuned the model since this time with additional experiments. It's not terribly surprising that the model could match an existing syllable production experiment. We use some of that data even to help define the locations of the model's components. But what's more interesting to us is now that we've got the model set up, can we generate a novel prediction and test that novel prediction and see if the model actually does what we say it does? Yes?
Can I assume that if you test 10 people, they would be more or less the same?
Well, this is an average of 10 people. If you've looked at fMRI data in much detail, single subject data can get kind of messy. So we model the average of 10 subjects in this case, or 15 subjects. And that's kind of necessary too, because the locations of our cells are kind of meant to be the average across the population. Any given individual has some variation in that, but on average, that's where you'd expect to see those. Okay. All right. So what I'm going to show you in the next part of the talk now is some predictions that we made in advance with the model once we got it to this form, and then tested with fMRI and also by looking at the productions of the model versus productions of actual speakers. And I'm going to start out with studies of the auditory feedback system. In particular, we've performed an auditory perturbation fMRI experiment, and this is what the experiment was meant to test. So recall from the earlier description that the feedback control system over here is comparing target auditory information with incoming auditory information, target somatosensory information with incoming somatosensory information, and it's detecting errors, and if there are errors, there'll be a corrective motor command. Well, once the system's tuned up, once we've learned the feed-forward command, it actually doesn't generate errors anymore, so these activities go down to 0 in the model. And in fact, these are areas of the brain where normally in a simple speech production task you don't see activity there. It's only when you do manipulations of speech that you see that activity. And our prediction was that, well, if we perturb auditory feedback, for example, then these auditory error cells should light up, basically. They should detect error. And what I mean by perturbed feedback is we have this subject saying words like bet, pet, and so forth, words with the vowel in it. On 1 out of every 4 trials, we'll take the first formant frequency in the and we'll shift it upward or downward. We built a DSP board that does this in real time, so it takes about 16 milliseconds from the time the speech comes in until the resynthesized perturbed speech comes out. The subject can't notice that delay, and in fact the delay's there whether or not we perturb speech, so they don't notice anything different, but on select trials we can make the feedback mismatch what they were trying to say. And when we do that, that should activate these auditory error cells, and that should lead to a corrective motor command. And so that's our first prediction here, that if we perturb auditory feedback while a person's speaking, this should lead to auditory error cells becoming active and motor corrective cells becoming active during the productions. So that's what we did. We did an fMRI study where we set this up in the scanner. It took us about 2.5 years to get this all working. There's a lot of issues with putting headphones in the scanner, putting a microphone in the scanner, and getting both headphones and microphone together in there and so forth, and getting the perturbation working. It was quite a technical challenge, but eventually we did get it working. And what we did was we shifted F1 either upward or downward on 1 out of every 4 trials by 30%. And that 30% is just about enough to make S sound like an A or an I, depending on which way you perturb it. The subjects actually don't normally notice this perturbation. We do it, they don't actually hear it. Here's what it would sound like. So here's an example sound before the perturbation.
Pet.
And this is what it sounds like after an upward shift of F1.
Pet.
So it sounded like pet the first time, it should sound more like pat the second time. The subject doesn't really notice this, but it turns out their brain does notice it, and we know that because we can see the activities, and we can also see that they correct for this perturbation, even though they don't consciously hear it. This slide shows the auditory results, or the acoustic results. This is a normalized version of F1. So if they produced bet normally, F1 would stay on this line here of a value of 1. If we shift F1 downward, the subjects actually compensate, and this gray area here is the subject's 95% confidence interval. What you see is that they compensate by moving upward. So they compensate in the direction opposite of the shift. That makes sense. They're trying to make it sound right. And what you see here is this line here is a simulation of the DIVA model with the same perturbation. And you see it falls within the confidence interval here. And similarly, if we have an upshift of the first formant frequencies, the subjects compensate by moving downward, and the model also compensates by moving downward. And another thing to note is that this compensation starts to take place around 100 milliseconds, which is what we predicted from the delays that we calculated from motor cortex to the sound output, from the sound output back up to cortex, and so forth. So we get the behavioral results matched by the model here.
This was a question I was going to ask earlier, but since now it's more important to understand how did you put these delays in the model?
How did we put the delays?
Yeah.
Well, it's a neural network where we have transmission from one.
So you really, they're built in? They're
Yes.
Okay,
Yeah. No, we built them in based on physiological findings. So we looked up how long it takes a sound to pop up in auditory cortex and so forth. Okay, so behaviorally then, the model accounts for what we see in the speakers, this compensation. This slide shows the fMRI results where we compare shifted speech to non-shifted speech in a subtraction. And so what we should see here is areas that become active due to the shift itself. And the prediction is shown in the bottom. This is a simulation of the model that we ran before the experiment. You can see we're predicting this auditory air activity here on the posterior superior temporal gyrus and in the planum temporale, which you can't actually see here, but inside of the fissure here. And in fact, we do get auditory activation here when we do the actual subtraction. So this prediction is upheld, and actually our locations are quite good here. And then we also predicted these motor corrective responses. But in the model, originally, we had the corrective responses going to motor cortex bilaterally. And what we found in the fMRI data is that it's actually right lateralized, and it goes to the premotor cortex rather than the primary motor cortex. So it looks like in this case that feedback control based on the auditory signal is right lateralized in the premotor cortex. The left hemisphere appears to be primarily involved in feed-forward control, so reading out commands from memory. The right hemisphere is more involved in correcting based on detected errors from sensory information. So this was a new finding. This was something that we used to modify the model. And the new version of the model that I've been showing has this right ventral premotor cortex feedback control map as a result of this finding. Okay, well, we also predicted that the same thing should happen in the somatosensory system. So in this case, we need to somehow perturb the articulator somatosensorily, meaning block the jaw or move the lips while you're speaking. And we need to do that in the fMRI scanner. That's another technical challenge. So we designed a machine right here that we call the perturbatron. And what this machine does is it takes a computer input, and every now and then the computer will trigger it. And what the trigger does is it drives this push-pull solenoid, which pushes air out through a long tube. This is like a 30-foot tube that goes from the console room into the scanner. And at the end of that tube is a little balloon that we've cut out that's shaped just to fit right in the mouth and kind of sit there comfortably normally. Normally the balloon's completely flat. It's made from the finger of a glove, in fact. So it lays in there pretty flat, and then every now and then somebody's saying ah, B, and during the ah, we blow up the balloon. It keeps the jaw from closing and perturbs their speech. And the perturbation can be turned on very rapidly within 100 milliseconds and shut back off within 100 milliseconds. So we can do a finely timed, unexpected perturbation of the jaw. And this is what we use. I'm going to skip over the details of the fMRI since I don't think there are too many imaging people here, and just show the results. Again, here's our model predictions. You see these error cells. Now they're in the supramarginal gyrus and the ventral somatosensory cortex. And when we do the shift minus no shift fMRI comparison, we get activity in that area where we predict for the error cells. But again, we get right lateralized frontal motor activity rather than bilateral motor activity. Again, this was the old version of the model with the bilateral motor activity. It's right lateralized, and that is why we've added in the past couple of years this feedback control map in the right ventral premotor cortex. Both for auditory and somatosensory feedback control, it appears that we're using this right ventral premotor cortex area right around here to help us correct for sensory errors that we detect, and that is dissociated from the feed-forward system in the left hemisphere. Just to note that if you get damage to your left ventral premotor cortex, you are left with what's called apraxia of speech. This is an inability to access or read out the motor programs for speech despite having no muscular problems. People can't read out syllables properly. That only happens with left hemisphere damage. That's consistent with the model here because only the left hemisphere has the feed-forward commands. Those are the ones you need. If you damage the right hemisphere after the model's already learned, it doesn't affect its output except in cases where you might perturb the auditory feedback because it's already got a well-tuned feed-forward command, and that's in the left hemisphere. It remains undamaged when you damage the model's right hemisphere. So this is one example of the model being consistent with lesion data on speech as well, and there are a number of others that we use to define the model. Okay. So to summarize then, these perturbation studies show that areas involved in sensory feedback control, these error signals are generated. We have locations where that happens. And then there's this right feed-forward control map, which we learned from our experiments where we tried to test predictions from the model. Okay. All right, well, that was a whirlwind tour of the feedback control system and a couple of experiments to test that. Next, I'm going to look at an experiment that looks at the feed-forward control system, and this is a different type of fMRI experiment, which uses an adaptation paradigm, which is something I'll describe momentarily. So in the DIVA model, the speech sound map cells are these cells here in the ventral premotor cortex and Broca's area. They have projections to the primary motor cortex, which are the feed-forward commands. And then there are also projections in the model that go through the cerebellum and then back up to the primary motor cortex. So there are two pathways from the ventral premotor cortex to the primary motor cortex. And one of the predictions we made in the feed-forward control subsystem in the model is that we said that these cells in the left ventral premotor cortex are involved in learning syllabic level chunks that have co-articulation built into them. So this part is responsible for learning an entire syllable from smaller motor commands, and so these cells are specific to particular syllables. And another way to describe that is to say that that part of the brain, according to the model, uses a syllabic representation. Well, can we test that? The answer is yes. There is a way to test it. Part of the problem with testing things like this is that fMRI has this slow delay, and so you can't really look at things differentially that are happening at a fast time scale. Instead, you get a picture of all of them at once. You can't see which one was activated first or not. That makes it difficult to kind of track through the stages. But there is a paradigm that allows us to look at what kind of processing units a particular brain area uses. And this is the fMRI adaptation paradigm. And it's based on a fairly simple finding. And that is simply that if we repeatedly provide a stimulus that activates a cell over and over and over again, that cell becomes habituated. It fires less and less and less. And it's been shown with fMRI that this also happens, that if an area is repeatedly doing the same thing, the same cells are being activated, you get less activity than if different cells in that area are being active. This is called adaptation. And what it suggests is that if we choose our stimuli appropriately, we can find out what each neuron or what each part of the brain considers to be repetitive. So one part of the brain might consider the same syllable to be repetitive if you produce it over and over again, but another part of the brain may only look at whole words and not really care about particular syllables, so that wouldn't be repetitive in that part of the brain. And I'll flesh this out a bit, but basically what we can do is design our stimuli in several different conditions that have different, the different conditions have different numbers of repeating syllables and phonemes and so forth. And by doing this, we can predict a pattern of activation that we should see in a region if it uses a particular representation. And I'll talk about that on the next slide. Okay, so this hopefully will make it a little bit clearer. Here I've schematized 4 different production conditions that the speakers have to go through. And down here I showed kind of what we expect for overall activity. A large activity bar here is what would occur if different cells are being used on each production attempt. And a small one is what would occur if there was adaptation, if the same cells were being activated every time. Well, the first condition is what we call the identical condition, and in this condition, the subjects read 6 words from the screen, and it's always the same word 6 times. So these are nonsense words. I should also point out these are French subjects. We ran this on, so this is a study of French language. And in this case, they would say pi gras, pi gras, pi gras, pi gras. And so the word level is adapted because it's the same word level stimulus, so we have very little word level activity predicted. The syllable level activity is also quite low for this because it's the same syllables over and over, and the phonemes are the same phonemes, so the phoneme level, everything would be expected to adapt. Phonemic, syllabic, or whole word representing cells all would be expected to adapt. The second condition, we just reorder the syllables, so ziklo, close, and in this case, the syllables and phonemes repeat, so they're adapted, but we have 2 words instead of 1 word, 2 whole words instead of 1 word, so cells that cared about the whole word wouldn't adapt fully. They'd have a little bit more extra activity. The next set of stimuli are things like low fu, blow, and these are stimuli that are chosen to force a resyllabification from one word to the next. So it's the same phonemes, but they're different syllables and different whole words. So the phonemes should adapt, but not the syllables and not the whole words. And then in the final condition, the variable condition, they read 6 completely different words with different phonemes, different syllables, different everything. And so in this condition, we'd expect everything to have a high activity. Okay, so how does this help us? Well, let's say we run all 4 of these stimulus sets, and for a particular brain area, we'll have a pattern of activity across those 4 steps, and we have predictions here. So if it's a phonemic representation, the pattern of activity across the 4 conditions should look like this. If it's syllabic, it should look like this. And if it's a whole pseudo word, it should look like that. Okay? All right, so we did that experiment. We had them produce those words, and what we found, the biggest finding for us at least, was that there was only one area in the entire brain that showed the syllabic pattern of activity. Here's the actual activity pattern. We had a pretty strict statistical criterion for whether or not it matched a pattern, so only very good matches were allowed here. And as you can see, this is an excellent match to the syllabic pattern, and it's in the left ventral premotor cortex, which is the area where we said we have these syllabic level feed-forward motor programs. So that was what we predicted. We were able to verify that. That's the first time that I think people have shown that this part of the brain uses a syllabic representation. Interestingly, we found phonemic representation in a number of areas, most notably the left supplementary motor area. The supplementary motor area is another type of premotor cortex. So we have the ventral premotor cortex and supplementary motor area, both of which are higher order motor cortices. We assumed in our model previously that they used the same sort of representation, that they were probably both syllabic, and that the SMA was simply activating a syllable motor program. Well, that's not the case. Actually, the SMA appears to use a phonemic representation, while the ventral premotor cortex uses this syllabic representation. This is something we're still trying to build into the model, but it seems to suggest that the SMA, which is involved in initiating motor programs, it has a lot of input from the basal ganglia. The basal ganglia presumably are telling the SMA when exactly to instigate the next motor command. It seems to be sequencing through phoneme-sized chunks. We think it's probably more closely related to articulatory gestures than phonemes per se, but it's a representation that there's one cell for each type of phoneme, not for each syllable. Those are being activated, and the ventral premotor cortex, on the other hand, is somehow stitching all of this into a well-formed syllable that has the proper amount of co-articulation in it, and so forth. And so we have this kind of mixed representation of the phonetic units in the two different premotor areas. And that's the main finding from this study.
So you just alluded to the fact that the articulatory gestures may be different than phonemic gestures.
Right.
I mean, what's
Right. So each phoneme has at least one or more gestures associated with it. And depending on how you define your gestures, you could say that each vowel has a unique tongue gesture, for example. And there, in a way, you could be like a one-to-one chord. So we can't really tell the difference between phonemes and gestures is the bottom line. But we can tell the difference between syllables and phonemes. And right. And well, and some of them may be built in. We're not sure. My reading of this is that we probably have primitive phoneme-level gestures that are old. The SMA has them very early on in our development, and that we're probably using much more of this ventral premotor cortex circuit to learn new syllables and to encode the information needed for the production of those syllables.
Would the model predict adaptation in the ventral premotor cortex for repeated syllables that are not identical, but which are composed of similar features worded in the same way? So if I just, for example, change the voicing feature of
Right.
nonsense, would you expect adaptation?
I would expect that would show significantly less adaptation, actually, as long as it's I mean, if they're similar, I suspect that there is some overlap in the motor program. So if it's only one feature that differs, then it's probably going to be adaptation, but not as much as if they were totally identical. That's my guess.
Would one example, for example, be to take alternating syllables where it's the same syllable, it's just stressed or unstressed? Meaning take two words which differ only in the stress, which are syllabically identical, except one of them has a stress on the first, the other one has it on the second.
I'm not sure what's going to happen there, actually. We've looked into, so stress control seems to be something that's built on top of the, like it's almost like you've got a one-dimensional parameter that you can stress it and then unstress it on existing motor programs. I'm not sure if that's something I don't think it's built into the motor program. I think it's probably something that is controlled externally, and if that's the case, then we would expect adaptation here with stressed and unstressed versions of it. It's an interesting thing that we may actually want to look at some point. Okay. All right, so bottom line here, we found different, and this just maps out the different type of representation we found in different parts of the brain. Noticeably, most of these things are going on in the left hemisphere where we got these clean sorts of results. The right hemisphere showed a Bessier pattern in general, except for the primary auditory cortex. But the main finding, again, is that the ventral premotor cortex has a syllabic representation. The SMA, on the other hand, has and the pallidum, which is part of the basal ganglia here. These are kind of highly interconnected. They use a phonemic representation. And the primary motor cortex actually doesn't care at all about phoneme or syllable identity. It just cares about overall amount of motor activity, it appears. And so we actually get a completely flat pattern. All the stimuli have the same amount of activity in the primary motor cortex. So it appears that for speech production at least, the interactions between syllables and phonemes kind of converge on the primary motor cortex to produce the overall output. Okay. All right. The last thing I'm going to do now is talk about using the model to investigate communication disorders. In some sense, this has been our main goal from the beginning. Our funding's from NIH, and the National Institute for Deafness and Other Communication Disorders. They care very much about disorders, and really what we're trying to do is understand the basic circuit so that we can figure out what happens when this circuit breaks. We've got a large number of collaborations going on looking at various types of disorders or impairments in speech. So, for example, with Joe Perkel and Harlan Lane and colleagues, we're looking at the speech of hearing impaired individuals, so people who went deaf after they learned to speak, and then they got a cochlear implant. We'll look at their speech before the implant and then after the implant and look at how they learn over time and try to relate that to the learning processes in the model. We've looked at acquired apraxia of speech. This is what happens with the left ventral premotor cortex damage, where you lose these motor programming abilities. That's largely what happens in the model. Childhood apraxia of speech, which is actually a bit of a different disorder, whereas acquired apraxia of speech involves a focal lesion in the left hemisphere ventral premotor cortex. Childhood apraxia of speech is usually due to things like lack of oxygen during birth, so there's kind of a general degradation in brain performance across the whole brain, not just in one particular region. Well, we can model that by adding noise to all the neurons in the model across the regions, and that produces different behaviors than the acquired apraxia of speech case. So we've done some modeling of that. We've done quite a bit of work looking at stuttering. Here's an example of a stuttering version of the model where we've made the feed-forward commands impaired. We've weakened the feed-forward commands in the model. It has to rely more on feedback control, and so it creates fairly large errors, and it resets and has kind of a stuttering-like behavior. Let's see if I can... So we can generate actual acoustics and kinematics from these impaired versions of the model. Then we can compare them to the behaviors that we see in people who stutter. For stuttering, for example, there are many different things that we can do to the model to make it stutter. So we use insights from existing neurophysiological studies of people who stutter. For example, a number of studies have shown white matter abnormalities underneath the ventral premotor and primary motor cortex. These, according to our modeling work, are projections that go down to the basal ganglia to give context about what's going on in the motor cortex. Well, we can damage those in the model, and that leads to a particular kind of stuttering, and we can compare that to the stuttering of people who have this sort of white matter problem. We are looking at people with amusia or tone deafness in collaboration with Gottfried Schlaug at Beth Israel. Ataxic dysarthria due to cerebellum damage, we're looking at with a couple of collaborations, Christy Spencer and Jeremy Schmahmann at Mass General Hospital. And the thing I'm going to talk about in the last part of the talk is a relatively recent project we've undertaken, started a couple of years ago. And this is a project on neural prosthesis, and this is done in collaboration with Philip Kennedy from Neural Signals Incorporated, which is a company that specializes in building neural prostheses. All right, so this neural prosthesis project starts out with Philip Kennedy's work in the late 1980s, where he developed an electrode shown here that's called a neurotrophic electrode. It's a particular kind of electrode that's got a glass cone at the end with 3 gold wires. This cone is less than 1 millimeter long, so this is blown up quite a bit. And inside of the glass, you put neurotrophic substance, which is a growth factor for neurons. And what you do is you push the cone into the cortical surface, and the open end, it's open on both ends. This pointy end ends up around layer 5 of the cortex, where we have the large output neurons from the motor cortex. Neurites grow in, so axons grow into this cone, and they stream through it, and they contact these wires, and they lock the thing into place. So you can permanently install this electrode and get signals from it several years after it's installed, which is way, way longer than most any other kind of electrode. Other electrodes are generally kind of externally mounted, and then they're pushed into the cortex. Well, the brain moves around relative to the skull all the time, and this causes micro movements around the electrode that changes the neurons that you record from, and it also causes scar tissue to build up around the electrode, and you lose your electrical signal after a few months usually. This electrode does not have that problem. It's made for permanent implantation. It was originally implanted in rats and then monkeys, and then in 1996, Phil Kennedy implanted the first human patient with this. And these are patients that have what's called locked-in syndrome, which is complete inability to move, so complete paralysis while being fully conscious and having all your sensory information coming in. So you can hear, you can feel, touch, you just can't move at all. It's actually a very hellish condition. And one of the main problems with this condition is there's no way to communicate with the outside world other than possibly slow eye movements. Sometimes there are residual eye movements, and so people can answer yes/no questions, sometimes by moving their eyes up and down. but they have essentially no ability to converse with people, anybody really in the world because of this problem. So they're willing to have these electrodes put in an attempt to try to decode their speech to send to a computer, for example, to produce speech output. And that's where we came into the project. There's a patient that they have there that was implanted in 2004, so he's had this electrode for 5 years now. And we took a look at the signals that were coming out of the electrode in our study. The first thing we did is we went into the images that we had from the patient, the CT images, to determine exactly where that electrode was, because for us that makes a big difference. Our model has different cell properties for different parts of the motor system, and so we needed to know exactly where it was in the motor system in order to have an idea of what to expect there. And we found out that this electrode is actually on the precentral gyrus at a location that's very near the mouth representation of primary motor cortex. It's actually right at the border between premotor cortex and primary motor cortex. It's on the crown of the precentral gyrus. The front part, or the back part of the precentral gyrus is primary motor, and the front part is premotor. The electrode's kind of right at the boundary between those two in the left hemisphere. Its location was determined using fMRI. They did a pre-surgical fMRI where the patient imagined producing words. The area that was on the ventral premotor cortex, or on the precentral gyrus, this gyrus here that was active during this task, was the area that was chosen for implantation. Okay, well, this procedure was done 4 years ago, and yet they were having a very difficult time trying to decode these signals coming out, and the one big reason is that there's no animal model for speech. So for arm movements or cursor movements on a computer, there are many, many monkey studies that show what the cells in motor cortex look like, what their properties are like, how they're tuned, and so forth. There aren't such studies from speech because only humans speak, and so we didn't really have an animal model to go by. And so the research group prior to our involvement were looking for things like whole words being represented by neurons, and there's nothing anywhere close to that clean cut in there. Instead, we've got about 40 neurons that we're recording from, and there are very subtle changes in their activities when the person is trying to speak. And it's the pattern across these 40 neurons that gives you information. Any single neuron doesn't tell you much at all. And what we decided to do was, in our model, the electrode is really on this pathway right here, from the ventral premotor cortex to the primary motor cortex. Well, in our model, this is involved in taking auditory trajectories, as represented by these speech sound map cells, and translating them into articulator trajectories. And it turns out that formant frequencies, which we use for our auditory representation, they're also quite closely related to the positions of the speech articulators. The tongue front-back position, for example, correlates closely with F2, and the up-down position with F1. So formant frequencies were what we determined based on the model is probably the best thing to look for here. We're in between ventral premotor and primary motor, going from auditory to a motoric representation. Well, formant frequencies are kind of in between those two. They're actually a very nice sort of representation to look for. And on top of that, we can pull out a two-dimensional formant signal that can be used to synthesize speech. And let me talk a little bit more about that in a minute. So I don't know how many...
Quick question.
Yep.
You said that there are no animal models for speech production. What about, there are these monkeys which have a fair number of vocalizations, maybe a dozen or more.
Well, monkeys don't learn, they can't learn new sounds. So they have, for the most part, they have a kind of built-in repertoire of calls, and they can choose between them, but they don't learn new ones. So for example, birdsong is more like human speech in one sense in that they learn a pattern, and then they can produce a novel pattern of their own. But neither of those systems are anatomically similar. Well, the birdsong is just very, very different. The bird brain is very different than the human brain. And the monkey brain, they don't learn new motor, auditory motor abilities with their vocal tracts, so they don't really have that system built in. So we wouldn't expect too many similarities.
Being able to choose from 12 vocalizations isn't quite the same task as...
Right. So in this case, you actually have to shape your movements to match an auditory target. They can't do that, is bottom line. Okay, so this is what the formant space looks like, a schematic of F1 versus F2 here. And I've plotted several vowels in this space just to show you that vowels can be represented as different regions within this two-dimensional formant frequency space. And what we're actually trying to decode from these the electrode, which is in these in this premotor / motor area, are formant frequencies. In particular, we're trying to decode F1 and F2. And so what we do is we give the patient a vowel sequence that he's supposed to try to imitate in his, he's supposed to try to say it. He can't actually move, but he can try to say it. So the sequence might be something like ah-ee, ah-ee. And we know what the formant frequency trajectories should look like for that. We have the cell activities while he's trying to produce those formant frequencies. And so we train a decoder to decode formant frequencies from the cell activities. And before we did that, we showed that, in fact, there is information about the formant frequencies in here. We can tune up, for example, that decoder on half of the stimulus and have it predict well above chance the other half of the stimulus. The fits aren't perfect by any stretch of the imagination, but they're well above chance. So there is formant frequency information in there. And so we set up a decoder to decode that. And the idea is that we can do this in real time. So as he's trying to produce a formant frequency trajectory, we're decoding those signals, and we'll send the formant frequencies that we decode to a formant synthesizer, which gives him immediate auditory feedback. So in the full system, which is schematized here, we're pulling out the information. It's a wireless system that sends the signals across the scalp. We do spike sorting to determine the neural site of origin. We send this, the spikes to a neural decoder that's tuned up to predict formant frequencies. Those formant frequencies go to a formant-based synthesizer. We're just using a CLAT synthesizer here. And that allows us to give the subject real-time auditory feedback. And the delay from these neural firings to the corresponding sound output is 50 milliseconds in our system, which is actually pretty close to the delay in an actual human brain, because it takes about 50 milliseconds for things that are activated in motor cortex to make their way out and to turn into sound. So this is quite a realistic delay for them. He's getting feedback that is roughly at the right time compared to if he was actually producing the words. I'll skip that. I'll end by basically showing you the results of the study. Over here is kind of tough to see, but this is a video of the actual setup. So here's the patient, the locked-in patient in his wheelchair. This is Dr. Kennedy working with him. This is our computer over here that's showing us what he's doing, and there's a synthesizer going on. In this paradigm, what we'll do is we'll give him a sound sample to imitate, which will be a vowel-to-vowel sequence like E. In that case, what would happen is that the computer would play E, and then it would say listen, it would play that, and then it would say speak, and then we turn over control of the synthesizer to him, and he tries to repeat that sound. And so he's trying to say and he's trying to move the synthesizer over here to the E target. That's his task. That's like a center-out task for those of you who are familiar with motor control. And here, what I'll show you in this video is 5 consecutive trials of production that show him. So each one will start with the target. So it'll say, listen, E, and the target will light up. It'll be the green one, and then it'll say speak, and then you'll see his attempt to produce it. So that was his attempt. That one was a very rapid success. In this case, he goes off, but he's able to kind of steer it back because of the feedback. Another good one. Next one's a complete failure. So you can see him kind of sampling around, trying to find how to move it in that case. So another success. So that's the state of the system right now. So he's able to hit the targets 70-75% of the time, And we've looked at what happens over time as he practices this, one of the most important things from our perspective is, can he use that feedback to tune up his brain so that he makes better productions with time? And the answer is yes. So here are slides that show as a function of blocks. So we ran 25 sessions across several months, and each session we broke up into blocks, and what we've plotted here is performance on the first block, second block, third block, and fourth block. This is the hit rate, just whether or not he eventually got to the right vowel. He has 6 seconds to do it. This is the endpoint error in formant frequency space, so in hertz. And this is the movement time. And you can see that, in particular, it's clear for this endpoint error that with each block, his performance gets better and better and better. So over the course of about an hour of practicing, he makes 5 or 10 practice attempts for a vowel. He gets much better in that time period. He goes from about less than 50%, like 45% correct, to above 70% correct. The way the system works now, we actually have to give him a new decoder every day, and so he has to retrain every day to use it for reasons that I'll be happy to talk about in the question and answer period if you want. But despite this, if we look across days and we look at his production, so we look at the endpoint error across the 25 sessions, we see that that is also decreasing. So he's learning something about the task that makes it easier for him to do it each day. It's unfortunate that right now we're not able to give him the same decoder, because our expectation is if he has the same decoder every day, then he should start where he left off the previous day, and that would give him a much higher ceiling of performance over time. Okay, so just to summarize the last part of the talk then, based on our model, we were able to come up with a decoding scheme that allowed us to translate these cell activities into intended formant frequencies, and this allowed us to build the first real-time speech-brain-computer interface. It's also the first time that somebody's used wireless telemetry to translate the neural systems for a real-time control system. The wireless telemetry is important because if you have an actual connector coming out of the head, the main problem with that is that infections start happening near the connector. In this patient, his head is fine. He's got hair. You wouldn't notice that he's got anything going on. There's no connectors or anything. When he comes in the lab, we just glue some coils at the right place on the head, and those pick up the signal. So we avoid the problem of having an actual connector going through the skin. And finally, this is the first example where an electrode has been in for 3 years and was still capable of doing real-time control. So all of these are very important steps toward developing real usable prostheses that are kind of permanent replacements for, in this case, the vocal tract, the ability to control the vocal tract. We're giving this person really a prosthetic vocal tract that they can use to just create speech output. The system needs a lot of improvements. One thing is that right now the decoder is just tuned up and then we just leave a fixed decoder. Well, there are techniques you can use where you can actually modify the decoder slowly with practice and help the learning occur more quickly. This is one thing we're implementing right now. There are better decoding techniques than the ones we've used. We used a very simple decoding technique. We used a Kalman filter to decode based on the firing rates, what the formant frequencies were. Another important step is to move toward a synthesizer that allows consonant production. Right now we have a synthesizer that makes vowel production easy, but consonants are very difficult to produce with that synthesizer. We're moving toward an articulatory synthesizer where he would actually be moving something like a virtual jaw, and that actually produces more realistic closure and opening of the vocal tract, and that leads to better sounding and easier to produce consonants, and so forth. Another major issue with our current situation is we have only one electrode. Well, it's got 2 channels in it, but that's not very much. We're planning to add more electrodes and smaller electronics so we can get more of that signal across the scalp over time. And I'll go ahead and stop there. I'll just leave my summary slide up for the model. Thanks. Yep.
Is there some way to use this technique to at least get a yes, no?
Well, that's the first. So we've been focusing on trying to get the speech output. We're setting up a collaboration with a company that builds augmentative communication devices, And what we want to do is use their they have these special laptops that are easily interfaced with and allow you to kind of use various control signals to move a cursor and so forth. We're planning to basically implement our system on that computer so that it can interact with the operating system. That should allow him to do simple tasks like that, like picking from one of 3 options. He can do that reasonably well now, but it takes a long time to build up that kind of system, and we've been focusing on just getting it to this point so far. Yes?
So you said something about, I guess it was right ventral insult to the brain after you've learned speech don't cause a problem because you don't need the error correction.
Right.
Is that good forever, or is there a limited amount of time that you can survive without having error correction?
Well, you get slow degradation. So the people who look at are people who became deaf as adults, and their speech remains intelligible for sometimes decades, but it sounds funny. I mean, they're acoustic, it's more nasal, for example, than because they don't have that system to kind of keep correcting for drift. Things drift into their productions that don't sound quite normal, but a lot of the motor program still seems to work pretty well, and they are more or less indefinitely intelligible, at least. Well, yeah, some things. There are things that happen immediately when you take auditory feedback away. First thing is that people talk louder immediately, and that does happen with these deaf patients as well. So some things change very quickly. Those we don't think are part of the motor program. We think those are more parameters that you adjust for every given production. You adjust the loudness level as you're doing it based on the environmental conditions. So we call those postural parameters, and we differentiate them from segmental parameters, which are more like the formants and putting your tongue in just the right place. The segmental stuff seems to last indefinitely. The postural stuff goes away very immediately, almost. Yes?
How much beyond the site of, because you had one slide, I guess, where the basic ganglia, there was a stroke, I guess the patient had a stroke.
Right.
What was the extent of the damage beyond?
Well, it was largely located to the ventral pons, and that's why it hit motor output, but not the so the dorsal column is where the sensory information is coming up. That is fine. It's this ventral part down here that was damaged. So it's fairly limited, but it's at a very crucial place, unfortunately. And this isn't the only thing that caused people to become locked in. Multiple sclerosis, for example, and ALS. ALS is a very common thing where it's a disease where you degrade pretty much until you die. It's fatal in the end, but there's a period of several years for many people where they become locked in, and yet they're still alive for several years. So some of the patients that have been used for this sort of study are ALS patients that then died within a year or two of implantation. The current subject we have is young and healthy except for the locked-in syndrome, so, and he doesn't have a degenerative disease, so he's somebody who could really benefit from this sort of technology, and he's also a great person for us to work with since he'll be around for a while. Yep.
Can I take the model sing a song?
Yeah, not yet. Have you seen Pava Robati, though? There is an articulatory synthesizer that sings songs. It's Ingo Tietze's model. It's called Pava Robati. Singing, in some ways, is actually easier to do than word production, strangely. Yes?
The additional electrodes that you're hoping to implant or planning to implant, what are your goals for those electrodes? You're going to do some type of mapping and then implant those electrodes as a result of that mapping. What are the mappings that you're...
So what we're planning to do is having the subject do several tasks in the scanner. One is just a jaw opening closing task. I mean, of course, they're not actually moving, but they're trying. Another is a lip task, and another is a tongue task. And then the final one is a speech task. And what we're going to look for are areas that are involved in both speech and one of the articulators. So if we find a jaw area that's involved in speech, we'll put an electrode there. That'll give us a lot of good jaw information. If we find a tongue area, we'll put an electrode there. That'll give us more tongue information and so forth. So the idea is to spread them out along the motor strip to try to get a richer set of information inputs by having areas that control different parts of the vocal tract. Does that make sense?
So one of the things that happens with the locked-in patients is that not only do they not have motor control, but I suppose because their jaw is not moving, they also don't have the sensory feedback.
Yes.
So how does that play into this?
Well, there's been a lot of wondering about how maybe you can't activate your motor cortical cells if you don't have the right sensory feedback as you're trying to move. However, there have been a number of studies that seem to show that you do still maintain the ability to activate the motor cortex for years after this sort of damage. So in our patient, we couldn't really, we could see motor activity when he tried to speak, for example. That's some degree of evidence, but other people have actually been able to decode that activity successfully and show that you can, control it to move items and so forth.
No, what I was thinking is, would you go one step further and stimulate these sensory parts?
Yeah, well, there's work going on in that regard. This is very difficult in our case because it's to know exactly what to stimulate, at least the way we've got it set up now with the formant synthesizer, is quite difficult. It's a one-to-many problem from formants to articulator movements. When we move to the articulatory synthesizer, it may be a little more reasonable to try to do that. I haven't seen results yet. I know there are people working on this for arm prostheses, but I haven't seen somebody that's successfully put in the feedback from a stimulator. I know there are people are looking into that, but I don't think it's...
Maybe just a prosthetic device that they put in their mouth, some sort of tactile haptic device, which then pushes their tongue or jaw to give them a feel of what their motor control would have done as interpreted by you.
Yeah, it's certainly possible, but that multiplies times 10 the technological problems right now. So we're basically asking him to be able to do this even though he's not getting the feedback back. We know that people can do that. He is getting sound feedback, so there is some, there's some bang for the buck with his movements. He can hear the changes, he just can't feel what it feels like. So, and just related, if you take a normal speaker and you anesthetize their tongue and everything, they actually speak relatively intelligibly until you do something like put a bite block in or make them adjust. So those feed-forward commands appear to be capable of being read out even if the sensory information is not coming in. But if you look at monkey deafferentation studies, what they find is that, they'll often deafferent the arms, meaning that they take away sensory information from the arms. And what happens is the monkey usually just doesn't move his arms anymore. He'll just keep them down there unless you make him desperate somehow, so he's super hungry and there's food, and then somehow he musters up the strength and figures out he can move his arm, and then actually starts to use it a little bit. This situation may be a little bit like that in that he's not getting the sensory feedback from his movements, but he is getting he's not getting the somatosensory feedback, but at least he's getting the auditory feedback, so, and he's able to do something with it. So.